Meiosis is a special type of cell division of germ cells in sexually reproducing organisms. It is used to produce gametes such as sperm or egg cells. Sperm is the male gamete, egg is the female gamete. 4 haploid daughter cells are formed from one diploid cell. Now, what is the meaning of this haploid and diploid? Now, this is a female germ cell. This is a male germ cell. Remember N. N is the number of chromosomes. And you know that we have got 23 pairs. Now coming back to this, whenever a cell gets ready for cell division, we have the DNA is duplicated, or it is doubled, or it is replicated. Therefore, N is the number of chromosome and duplicated, no? So 2 multiplied by N, you'll get 46. So this is a diploid cell. Similarly, even in the case of the male germ cell, 2 multiplied by N is equal to 46. Now these germ cells, they produce gametes, which is an egg in the female and a sperm in the male. Now, this diploid cell undergoes meiosis, and in meiosis, the number of chromosomes is half. So therefore, the diploid became haploid. You had 46, 23, here again 46, it became 23. So a diploid cell produces haploid cell. Now, when the male and the female gamete come together, that is the egg and the sperm, fertilization takes place and a zygote is formed. 23 pairs from the 23 from the female, 23 from the male, maintains the number 46. So the zygote is diploid. So remember, duplication, replication, copying takes place only to maintain the number of chromosomes. Now you will wonder why we have to maintain the number of chromosomes. Now if we have extra chromosomes, it will create problem in the young one or the baby. The baby will fail to develop properly. And one of the most common disease because of extra chromosomes is Down syndrome. That child is mentally and physically challenged. Another common disease because of less number of chromosome is Turner syndrome. Such a child has a short stature, ovaries are not developed, so she or he cannot reproduce, she cannot reproduce, and heart defects. Before entering prophase, the cell has already undergone interphase. Now what happens during interphase? This is a cell. This is the plasma membrane. You see this inside, this is the nuclear membrane. These are the chromatin fibers. This is the nucleoplasm. Outside the nucleus is the cytoplasm, and this is the centriole. We are not drawing all the cell organelles because what is important for cell division, we are focusing only on that. So now during interphase, the nuclear membrane is intact. The centriole is at the same place, there is no change. The DNA has duplicated, replicated, or been copied. The cell now is diploid, that is 4N. It has grown in size. The chromosome number is the same. Now also you'll find that the centriole over here has started splitting. The cell is now ready to enter the first subphase of prophase 1, which is called leptotin. Leptotin means thin threads. Now, when you look at the cell over here, we will find that the nuclear membrane is intact. The centriole is at the same position. There is no change, but it has split and not started moving. Then you'll find that the chromosomes have double DNA, that because they have replicated or they have been copied. And the chromosomes now over here have started condensing. Now this condensing is not continuous. It happens at regular intervals. That is why you'll find the chromosome over here have got beaded appearance. The cell now moves into subphase 2 of prophase 1, zygote. Zygote means paired threads. Now let us look at the cell in subphase 2 zygote. The nuclear membrane is intact. The split centriole is at the same position. There is no change. The chromosomes have condensed. They no more look like threads, but they give the appearance of the chromosome. And they have started pairing together. Pairing of homologous chromosome takes place. Now what is the meaning of the word homologous? Homologous means if this is from the mother, this one is from the father. So one comes from each parent. One comes from the father and one comes from the mother. So the pairing has taken place. And now these chromosomes, they have duplicated, they have replicated. These start splitting vertically. They split vertically and they look somewhat like this. They split vertically and they look like this. Now these two arms are called sister chromatids. These are the arms 1, 2, 3, 4. These are called sister chromatids. Even in this case, these are the two sister chromatids. But if you look at one arm of the first chromosome and one arm of the second chromosome, these are called non-sister chromatids. Now once they have paired, they have to be held together. Now how are they held together? They are held together by a protein network at different places. Just as you staple something, like that they have a network which is holding them together. And once they are held together, then what happens is crossing over will take place. They just cross each other and a shape like this is formed X. The cell has now entered subphase 3 of meiosis 1, patchy team, which means thick threads. Now the chromosomes have condensed. They are no more thin like fibers. Therefore the name patchy team. Now look at the cell. The nuclear membrane is intact. The centriole, which has split, is in the same position. It has not moved. Now crossing over started at the zygote phase And now the crossing over, remember, takes place between the non-sister chromatids, not between the sister chromatids, the non-sister chromatids. Now, in the previous phase, they just crossed over. Now the fusion of the crossing over has started, and this fusion is known as chiasma, where the genetic material is exchanged. This will result in genetic variation. Now this crossing over can take place at one place or two places, but the maximum number of places where crossing over can take place is 4. The cell now moves into subphase 4 of meiosis I, diplotin, meaning separation. Now let us look at the cell. The nuclear membrane is still intact. The centriole which has split is at the same position. Crossing over is complete. Now, once the crossing over is complete, they don't have to be together. So that protein which is holding them together slowly dissociates. And the chromosomes start moving away from each other. Now here you'll see the sister chromatids. Here there were 4 arms, 1, 2, 3, 4. These 2 arms have fused and one is formed. So here and here fusion has taken place. Only where they are cross, they are still attached. The cell now enters the last phase of prophase 1, subphase 5, diakinesis, meaning the final. Now look at the cell. It has changed from what we have observed in the other subphases. The nuclear membrane has disappeared. The centrioles have moved to the opposite poles. The spindle fibers have started forming from the centrioles. The chromosomes are clearly visible with the exchanged genetic material. You can see every chromosome has got light, dark, dark, light, light, dark, dark, light. Now why is this difference? That is to show that there is exchange of genetic material. And you'll find that the spindle fibers are attached over here to the kinectopore here. This is the part of the chromosome. And these spindle fibers are pulling the chromosome and trying to bring them along the equatorial plane. The cell now moves into metaphase 1. In metaphase 1, the nuclear membrane has disappeared. The centrioles are at the opposite ends. The spindle fibers are fully formed. The homologous pair of chromosome, homologous I told you, one from the father, one from the mother, with the genetic exchange, they are aligned on the equatorial plane. And I told you that the spindle fibers are attached to the chromosomes at the place with this red spot, which is known as the kinetochore. After they have been arranged at the equatorial plane, the cell enters the next stage, that is Anaphase 1. anaphase. Let us see what happens. Now the spindle fibers, the arrow is showing what the spindles, they are contracting. That means they are pulling the chromosome towards the opposite poles. So the chromosomes are moving towards the opposite poles. So that causes reduction in the chromosome, because 2 have gone this side, 2 have gone this side. So the chromosomes are reduced to half. Once they reach this pole, they enter the next phase, which is telophase. And also we will observe that in between the chromosome there are no fibers. In telophase 1, karyokinesis is complete, that is, nuclear division is done. Nuclear membrane reappears around the chromosomes. See, the nuclear membrane has started appearing. The chromosomes have reached the poles. Now they have become thin because they have started decondensing. The centriole is at the end. The spindle fibers have dissociated, they've disappeared. Now what is very interesting is, you'll find constriction starts because now the nucleus has finished dividing. Now over here the cytoplasm will divide. So therefore you will see this constriction. The moment you see this constriction, it is an indication that end of telophase 1. Now, in cytokinesis 1, cytoplasmic division will happen. I told you have in the previous case, constriction on the plasma membrane was seen. Now this constriction gets deeper and deeper, and from one cell 2 haploid cells are formed. That means from 2n you get n and n. So 2 haploid cells are formed. Now the number of chromosomes is half. Parent cell had 2n, that is diploid. The daughter cell has n, that is haploid. The centrioles are still at the original position. They have not moved. Now meiosis 1, in short, is reduction division. Once cytoplasmic division is complete, both the cells move into meiosis II. And remember, whatever changes happen in one cell, the same changes happen in the other cell also. Now the centrioles have moved. In the previous case, we had the centrioles here. Now the centrioles have moved here. Okay, so this at one pole and this is the other pole. So the centrioles have moved horizontally. In the previous case in meiosis 1, they had moved vertically. The spindle fibers have again started forming. And remember, these centrioles are responsible for the formation of the spindle fibers. Now this chromatin network of fibers, they start condensing. And you can see the chromosome with the exchanged genetic material. See, you can see a little darkened part over here and you can see a light part over here. That means there was exchange. And these spindle fibers are going to pull the chromosomes so that they are aligned on the equatorial plane. We have just discussed the changes that took place in the cell during prophase 2. Now the cell is ready for metaphase 2. Look at the cells very carefully. You can see the chromosomes have aligned themselves in the equatorial plane. They are fully condensed. The arms are seen very clearly with the exchanged genetic material. The spindle fiber is attached to the chromosome at the kinetochore. The cell is now ready for the next phase. The cell now is in Anaphase II. Look at the picture clearly. The chromosomes split. Now, that is the chromatids separate. All this time, the chromosomes were moving. Now, you know that a chromosome is made up of two sister chromatids. So, the chromatids separate. That is, one chromatid is the original DNA. The other one is the copy of the DNA. Once the chromatids have separated, the cell enters telophase II. In telophase II, the nuclear membrane reappears. Spindle fibers disappear. Chromosomes change to their thin fibers called chromatin. Karyokinesis is done, that is, nuclear division is done. Constriction appears in both the cells. It means the cell is now ready for cytoplasmic division, that is cytokinesis. Now remember, over here, each cell has received two chromatin fibers. So two here, there are two chromatin fibers here, there are two chromatin fibers here, and two chromatin fibers here. We have now come to the end of Meiosis II. That is cytokinesis 2. This constriction gets deeper and deeper, and from each cell, two cells are produced. So in all we get 1, 2, 3, 4 cells. Here they have separated. These are the 4 cells. These 4 cells are known as the daughter cell, and these daughter cells are haploid. That is, they have exactly half the number of chromosomes as compared to the parent cell. A summary of what we did. We have, just before a meiotic division, the chromosome number doubles. So the parent cell is now 4n because the DNA has replicated or duplicated or being copied. Now in the first meiotic division, which is known as reduction division, from the parent cell we get 2 daughter cells. So now 4n is divided into 2n and 2n. So these are diploid cells. Now these diploid cells undergo the second meiotic division and they produce 4 germ cells which are haploid. Now these 4 daughter cells have got 2 chromatin fibers. These are the gametes. That is, either they are the egg cells or they are the sperm cells. Now you know, for sexual reproduction, gametes are required. That is, a sperm cell and an egg cell will fuse and they will form a zygote, and the number of chromosomes is restored.